Hi there. Today we are doing chapter 16 of your textbook, Transplant Immunology. Um, we are going to, this is a little roadmap of where we're heading. If you're interested, we're going to look at we're going to pretty much cover this material today. Histocompatibility, different kinds of grafts, different mechanisms of rejection, which include kind of our typical adaptive immune response, as well as this kind of funky little response called graft versus host, which we're going to see is that the immune cells of the donor turn against the recipient rather than vice versa. We're going to look at some lab methods for typing HLA antigens and HLA antibodies, a little bit of molecular methodology for antigen detection, and then some discussion about immunosuppressive agents. Transplant immunology is, not surprisingly, the study of the immune response that occurs when an organ or tissue is moved or grafted or transplanted from one individual to another. A major problem that we see following transplantation is that the immune system of the recipient sees the grafted tissue as foreign. It attacks it and destroys it. And it destroys it through, kind of the traditional adaptive immune responses that we're used to seeing. But it also an immune response can be, again, as I said, where donor immune cells prompt an immune response against, recognize the recipient as foreign and prompt an immune response against them. And that's called graft versus host disease. And in evaluating these rejections, we're going to look at trying to minimize them. And we do that by making an assessment of the compatibility of the donor tissue compared to the recipient. And that process is called histocompatibility, histocompatibility testing. But the process is assessing histocompatibility, tissue compatibility, the tissue compatibility between donor and recipient. And that all starts with HLA antigens and the MHC complexes. So really the primary immunologic mechanism that we're going to talk about here is revolving around the specific genetics of an individual expressed through their human leukocyte antigens. You're going to see it in the book, you'll see it in today's text, you'll see it in things that you read online, articles, that MHC and HLA complexes are pretty much interchangeably used in terminology relative to transplants. It comes from the fact that they really are the same thing. MHCs, there's a version of MHCs in every vertebrate. In humans, we call that MHC complex the HLA antigens. But comparatively, in mice, the MHC is termed the H2 complex or H2. So all vertebrates have an MHC. The ones exclusive to humans are called HLA. The ones exclusive to mice are called H2 complex or H2. So hopefully that clears that up. HLA proteins or MHC proteins that we expect are involved. We know this. They're involved in antigen processing and presentation. We're going to see that they're coded by genes, by HLA genes that are closely related to each other within the MHC complex on the chromosome 6, the short arm of chromosome 6. We're going to identify these HLA antigens and the importance of them, and why the differences between HLA antigens on the donor and the recipient can cause an immune response that's going to ultimately lead to graft rejection. The HLA system is the most polymorphic of all our antigen systems. And so because that polymorphism, the HLA antigens, when they do elicit an immune response, elicit a nice robust one. It's going to be a good strong immune response. And so tissue transplants are dependent on managing the reactions between donor and recipient. And by having really close antigenicity of tissue types, we can hopefully restrict the immune response or dampen the immune response. That polymorphism, though, it really restricts the transplantation process if you're not a really tight match. And if you're not, you're going to be a long-term immunosuppressive therapy. And it's going to be a battle for a long time. So what do we do? One of the ways we do this, and I think that everyone probably has heard about this. I don't think it's a surprise to anyone that, when we're looking at transplants, we look at siblings first, not parents. We're going to look at siblings first because they're going to have the highest likelihood of having genetic makeup or haplotypes of HLA antigens that are consistent with the recipient, the potential recipient. So we're going to look at that as well relative to HLA. So, but first let's look at, the histocompatibility systems. There are several antigen systems that can stimulate graft rejection. The main system, as I just said, is the HLA system, or the major histocompatibility complex. We know this. We love it. We've read all about it in Chapter 2, and we've talked about it a lot. We keep encountering it in different discussions because it's a really important piece of the immune response. Last week we talked about it in reference to autoimmunity and identification of self and non-self, and misidentification of self, I guess, would be even better. We've talked about it relative to genetic diversity. And so we're pretty familiar with this, but let's take a few minutes just to look through it. And all I can say is, don't forget, if this is in Chapter 2, if you have any questions, go back, read it, feel comfortable with it. The MHC is going to be something that, we're going to keep talking about in the semester. So it's important that we make sure you're comfortable with this. As I said, the HLA mismatch, or the genetic mismatch of HLA antigens between the donor and the recipient, is what's going to cause ineffective or unsustainable grafts. It's a really important thing for us to do, and so we're going to be looking at those, and we're going to be matching them up. We're going to do our best match of all between the donor and the recipient, the most likely that we're going to be able to get in order to have an effective transplant. If we look at this picture that I think you're all going to recognize from Chapter 2, we talked about it, and we're looking at kind of the organizational structure of the MHC complex on, again, the short arm of chromosome 6. Here's the short arm, here's the long arm, and here's the MHC region. If we look at how these are organized, the genes on chromosome 6, let's take a look at how they're organized. So we have HLA genes here. There are 3 of them that are involved in code for class 1 MHCs. So we have 3 HLA genes, HLA-A, HLA-B, and HLA-C, and these all code for class 1 MHCs. There are 3 gene systems, or 3 gene regions, that code for class 2. These are all called the D region, but there's DR, a DR region, DQ region, and DP region. So there are 3 regions, all kind of, classified together as the HLA-D region. We have an A, B, and a C, and a D region, but individually there are 3 regions of genes also within this overarching D. that make up class 2. Class 3 MHCs we didn't talk about in class. Remember, this is really not related to what we're talking about in immunology, so we're gonna skip past that. As I said, those genes are all those HLA genes are all closely linked on chromosome 6, and it's that it's the genetics, inheritance patterns, and the coding of HLA antigens that create this really highly polymorphic or variable or allelic characteristics of the MHC that create that strong graft reaction that I just spoke about. So, and then I guess the last thing to say really about the MHC in a general way is that we're going to talk in a few minutes about how we inherit a gene, a haplotype from each parent to make a new gene for each individual. So we're going to see how we take individual haplotypes from each parent. So we'll look at the inheritance patterns of these genes in just a few seconds. So we'll look at that. Let's try and remind ourselves, though, about the functionality of MHC. The process of MHCs, the goal of an MHC or the function of an MHC, what does it do? It's designed to process antigens into small particles and then present them on the cell surface to recipient T cells. That's in general, right? So antigens can either be from within the cell or and those are called endogenous antigens. Things like viruses or even self-antigens can be endogenous antigens. Or things from outside the cell, exogenous antigens, things like bacteria that can be brought into the cell for processing. But in any case, MHC, the MHC complex, takes the antigen, it degrades it and puts it into little teeny pieces, the antigen presenting cell. Within the antigen presenting cell, that antigen is broken down into little small pieces called peptides. And those peptides bind into the MHC, and through slightly different processes between class 1 and class 2 proteins, the MHC brings the antigenic peptides up to the cell surface. We call those little peptides epitopes, right? They're the little antigenic piece or an antigenic piece of the antigen that was processed. And the MHCs present that antigen to an appropriate T cell. How does it know what T cell to present it to, and how does this process all work? Well, let's look at class 1 proteins first. So class 1 MHCs, as I said, were a result of gene coding in the HLA A, B, and C. So the genes HLA A, B, and C code for class 1 MHCs. Class 1 MHCs are all expressed on nucleated cells, so every nucleated cell in your body has MHC class 1 complexes. MHC class 1, as we remember, are involved in the processing and presentation of endogenous antigens, those ones from inside of the cell. And it's class 1 proteins actually have a structure very similar to immunoglobulins. And this is an A, it should say alpha here. I think it should have been a little A instead of an A, but whatever. Alpha heavy chain, so it has an alpha heavy chain, and that's where the polymorphism is. That's where the genetic variation takes place. So we have the alpha chain, which is the polymorphic area of the MHC, And then we have a beta-2 microglobulin light chain. So we talked in immunoglobulins, there were either kappa or lambda light chains. In class 1 proteins, they're beta-2 microglobulin light chains. And the purpose of this beta-2 microglobulin light chain is that it causes the cells with class 1 MHC proteins to only present their peptides, their processed antigens, to cytotoxic T cells, to CD8 positive cells. So the beta-2 microglobulin only allows recognition and presentation to cytotoxic T cells. So again, this is the polymorphic area of the MHC, and this is the one that selects the appropriate kind of T cell to present to. And that makes sense, doesn't it, that we would only want to bind to and have receptors that would bind with the appropriate T cell. If you think about it, cytotoxic T cells are going to be effective against things that live inside of a cell, like a virus, whereas an antibody is not going to be as effective while it's secreted away inside of a cell, right? So it's an important thing to be able to present to the right cell, and the beta-2 microglobulin does that. Class 2 molecules, on the other hand, are, the genes located in the HLA-D region are the ones that will code for MHC2 proteins or MHC2 complexes. This also has a polymorphic heavy chain, but it has, instead of a beta-2 microglobulin chain, it has a beta light chain. And the beta light chain is very much the same concept, but instead will only process and present to the appropriate CD4 positive T cells, those naive T helper cells. The purpose of the beta light chain is to, again, identify that the processing and the presentation has to go to a CD4 positive T helper cell. What else do we know about class 2 MHCs? We know that, and I apologize it's not on here, I'm not sure why. Unlike class 1, class 2 proteins are only expressed on antigen-presenting cells. What kind of antigen-presenting cells? Dendritic cells, macrophages, things like that, as well as something like a B cell would be a good antigen-presenting cell for class 2 proteins. So it's expressed only on APCs, and it is results in the processing and presentation of what? Exogenous antigens. So if you remember, you have bacteria or some other pathogen outside of the cell. It's engulfed in by phagocytosis into an antigen-presenting cell. A macrophage gets processed inside. The MHC presents it to T helper cells on the cell surface. And so, as I said, by restricting class 1 and class 2 molecules for recognition by and presentation to specific T cells, the MHC molecules are only going to interact with the appropriate lymphocyte, express the appropriate T cell receptors, and the ones that are going to aid in the identification of the specific antigen, one with a good antigenic affinity for the binding cell, and one that's going to be an effective immune response to the antigen. So it's a kind of an amazing little process, isn't it? As I said, it's important for us to remember that, as I said, this inheritance from each parent of the genetic material that makes up the major histocompatibility complex, the haplotypes, the genetic variation that results, comes from each parent and the inheritance from each parent. And so we're going to take 1 second to look at the Mendelian codominant inheritance patterns of HLA haplotypes. This is important because we've talked about how the gene variants of MHCs, or the alleles, are really polymorphic, and this allows for that strong genetic diversity. It's going to help the host define and detect self from non-self. This, in the end, is what ends up causing and resulting in a really strong immune response in graft reactions, as well as hopefully identifying self. And we have processes within this systems that are going to reject the processing of self-antigens and presentation of self-antigens for response. We're going to have inhibition reactions that will stop that. But you can see from this illustration from your book that you can, just based on, two parents, you can end up with really diverse haplotype inheritance among offspring. That's that genetic diversity that we talked about. We're not getting 4 children that all have the same genetic makeup. That would not be genetically diverse. Instead, we have genetic diversity here, and the more, obviously, that you have more parents that are having children and their children's children, you introduce more genetic diversity consistently. So let's look at the inheritance pattern of MHCs. It's pretty straightforward in that one haplotype is inherited from each parent, and if you do the math, the father here has 2, and the mother has 2, so it's the maximum number of combinations that can be made. If you put it in a Punnett square, you would have a 2 by 2 Punnett square, so you would have a maximum of 4 possible inheritance patterns for offspring from these 2 people. So in the book, they marked each haplotype as either A or B, or C or D for the maternal inheritance to the offspring. And that's, I think, just for ease of being able to monitor them as we go along. So as I said, there's only 4 options. The father is going to have, because we have a total of 4 potential variants of these 2 parents. The father will contribute haplotype A to 2 of the offspring and haplotype B to 2 of the offspring. So there's the A, the B, and the B. Similarly, the mother will contribute the C to 2 offspring and the D to 2 offspring. So there's C, and D, D. And so if we look at the genetic inheritance that came from each, the first potential offspring has a haplotype A from the father and a C from the mother. That's the combination there. The second one has an A from the father, a D from the mother. The third has a B from the father and a C from the mother. And the fourth has a B from the father and a D from the mother. No matter how hard you try, you're not going to come up with more than 4 potential offspring haplotypes. And if in this genetic pattern, feel free to go look it up and figure it out if you doubt me. But 1 haplotype is inherited from each one. So why is this important to us? When we're looking at transplant immunology, we're trying to figure out how close can we get that HLA pattern between donor and recipient. Remember how I said we look to siblings first? This is why. So at any given point in time, there's a 25% chance that 2 of the siblings are going to have the same identical inheritance of haplotypes from their parents. So if this was me and I needed a transplant and I was checking my siblings, um, there's a 25% chance that one of them will look exactly like my genetic pattern. So if I look, and I can look at the 4 examples here, there's only one of them that's an AC. So 1 out of 4 is a 25%. We call that HLA identical. There's a 50% chance that one of my siblings will be HLA haploidentical. And what that means is that we've inherited one haplotype of the 2 that I've inherited the same as each other. So if we look at this again at our 4 possible things, I'm an A and a C. There's one here that's an A. That's a D, so that's not a match. This one has a C, and I have a C, so that's a match. It's a haploidentical match. And this is a B and a D. This has no match to me. So this is non-identical. So 2 out of 4 will have one haplotype the same as me. So I have a 50% chance of being haploidentical, and then obviously a 25% chance of being non-identical. This, as I said, is going to be important when we're looking at potential donors from siblings. So if we look at question 1, what's the probability that any 2 siblings will receive the same haplotype from both parents? That is, to be HLA identical. We just talked about that, and we saw that 1 out of 4, or 25%, of siblings from the same parents have the likelihood of being HLA identical. So the answer to question 1 is 25%. So that was yep. So we talked about that there are several antigen systems that can elicit and stimulate a graft reaction. The first one was the major histocompatibility complex. We just went through that. It's hard to imagine when getting to the next. There are other non-MHC systems that can also stimulate a graft reaction. What are those systems, and what are they called? In general, we call these non-MHC systems the MHAs, or minor histocompatibility antigens, or the minor histocompatibility antigen systems. They were detected when it was discovered that siblings with HLA identical HLA antigens were the donor and the recipient of tissue transplant, and still had graft reactions, graft rejection reactions. And they knew it had to be something else. The HLA patterns and phenotyping were identical, so something else had to be causing that graft reaction. It wasn't the HLA antigens. So when they looked, they found a number of different minor antigens that can also cause rejection of tissue transfer. So what are they? Actually, what else do we know about them? They go at a slower pace, so they do cause graft rejection, but at a slower pace. They're a result of variations of amino acids on X-linked and autosomal transplants. And the graft rejection is very similar to what we would expect in a typical CD4, CD4 or CD8 mediated reaction. So it's a typical adaptive immune response, nothing fancy to it. It's not something like graft versus host where it's something different, but it's really a typical immune response. The 3 systems that we think of that are involved with these minor histocompatibility antigens, there's 3 of them. There's the MICA antigens, which are the MHC class 1 related chain A antigens. It's a mouthful, right? That's why we call them MICA. MICA antigens are the result of gene coding for cell surface proteins, not on T and B cells, but rather on some secondary kinds of cells like epithelial cells, dendritic cells, monocytes. So it's an MHC class 1 related antigen on other cells besides T and B cells. Not surprising that it's class 1. Why? Because we know class 1 is found on all what? All nucleated cells. So these are clearly nucleated cells, but we don't want them on T or B cells because this is those are the main cells of the immune system. So these are kind of a secondary class of antigens. They're not found on the main immune cells. What ends up happening with these? It's very similar. So, for example, maybe if you have a transplant, there are these secondary cells that are that are transplanted with the solid tissue, whether it's a dendritic cell, something that came along for the ride, monocytes or something, and antigenicity from the donor reacts with the recipient, and the recipient rejects, identifies those MICA antigens as foreign, and adaptive immune responses follow. These are MICA antigens are just another genetic variant of MHC antigens, class 1 antigens, and we see these typically in things like kidney rejection. They do result in decreased graft survival, but again, it's a slower process than MHC rejection. The second non-MHC system that's involved in graft reactions are the ABO group. The ABO groups. We absolutely do ABO blood group typing when we are looking at transplants. You can have blood group antibodies that are going to bind to, from the donor to the recipient, that will bind to the vasculature of the tissue or the transplant, causing an antibody-antigen reaction in the vasculature. What do we know that does? We know that activates complement, right? Leading to really rapid rejection of the organ or the tissue. ABO reaction, and again, this is just the antibody rejection by ABO antibodies, is termed a hyperacute reaction. It's a very fast reaction, just as if it was a blood transfusion reaction, very quick. Maybe not as not 100% as quick as that, but it can be. What can they do if they happen to have an ABO antibody that's reacting with the recipient? They may be able to do something like a plasma exchange, where they exchange out all of the transplant recipient's plasma, or the bulk of it. You never get all of it, but they do a large volume plasma exchange, and that eliminates the blood group antigens from the donor, the blood group antibodies from the donor, from the patient plasma. Similarly, what do you think they could do with an immunoglobulin administration? I think that if you think about it, you could have, if you had, say, a group B antibody that's reacting with patient cells with group B antigen on it, you could add an anti-human anti-B antibody. So we could bind the B antibody of the donor so that it can't react with antigens in the recipient, and that could eliminate that process as well. So there's a couple of actions that can take place to help with this minor histocompatibility antigen process. And the last system involved in graft reactions are called the KIRs, the killer immunoglobulin-like receptors. And these are interesting. They regulate the activity of natural killer cells. They it's in the form of, maybe easiest to explain it in an example. So the actions of killer immunoglobulin-like receptors, they're cell surface molecules that regulate natural killer cells relative to self and non-self. If the circulating NK cell encounters a self-antigen that was presented by MHC class I molecules, the KIR, the killer immunoglobulin-like receptor, causes an inhibitory response that keeps the natural killer cell from responding to a self-antigen, which is a good thing, right? We don't want natural killer cells identifying self for destruction, right? Conversely, though, if the natural killer cell comes in contact with an HLA protein that is not self, it's a non-self HLA protein, the inhibitory receptors that we just saw, the KIR inhibitory receptor, does not get engaged, and the natural killer cell becomes activated and kills that non-self, the cell with the non-self antigen on it. So KIRs are mediators of the actions of natural killer cells, natural killer lymphocytes between self and non-self. We talked about next, I guess now it's time to get into the business of transplants. We talked, what we've talked about so far was kind of the background of how we get where we're going. How do we classify the transplants or the grafted tissue? 4 major types of grafts, and the nomenclature is based on the relationship between the donor and the recipient of the graft. An autograft is a transfer of tissue from the same individual. The example we see in the book was the saphenous vein of in a cardiac bypass, but it could be something like a skin graft as well, self to self. When we get tissue transfers or tissue transplants from a genetically identical individual, that is an identical twin, it's called a syngenic or an isograft. So that's just, identical antigenicity. Genetically identical, oh my gosh, genetically identical tissue. It was tough to say 3 times fast. Grafts between non-identical individuals of the same species is termed an allograft. So that's just human to human. Maybe we get it from a cadaver. Maybe we get it from a stranger. We get it from an organ bank. We get it from wherever. Maybe it's a piece of a kidney, a piece of a liver from a relative or something. Non-identical though, genetically not identical. This is the most common kind of transplant that we have, allografts, human to human. But there are also transplants between members of different species, like pig valves in humans, and those are called xenografts. And both xenografts and allografts, their process by which the rejection of their grafted tissue happens is really by something that we term allorecognition. And what allorecognition, we're going to see 2 examples of that in just a moment. Allorecognition is the first step in a series of events that ultimately leads to some form of T-cell activation. May cause antibody production, but ultimately it's designed to reject grafted tissue. So let's look at a couple of different mechanisms by which allorecognition can occur. And this will be very familiar to you. This is our typical adaptive immune responses, but this is in reference to transplanted tissue rather than pathogens that have entered the host. As I said, most human transplants are allografts. The recipient's immune system recognizes the foreign HLA proteins on the graft cells by one of two mechanisms. These are the two mechanisms. They're going to look super familiar to you because we've seen these repeatedly. These are two of the mechanisms of the adaptive immune system. So let's take a look though. Indirect allorecognition, not indirect, but indirect allorecognition. Cytotoxic T cells from the recipient recognize and bind directly to foreign donor HLA antigens on the cells. The T cells from the recipient, it's hard to keep doing this donor and recipient thing, I apologize. The T cells from the recipient release cytotoxic factors to destroy the grafted tissue. Pretty straightforward. The MHC, we are going to see MHC molecules on antigen presenting cells that are going to recognize this, bind it, and cytotoxic T cells are going to release their cytotoxic factors to kill it. In the lab, we can use the principles of direct allorecognition to determine if donor tissue is likely to stimulate a graft rejection. We call this the mixed lymphocyte reaction. It's a test. It's an in vitro test. And the way that it works, I'll explain it to you. It's in the book as well, I believe. It is in the book. We are going to, in the test system, we're going to incubate the lymphocytes of the potential recipient with some inactivated lymphocytes of a potential donor. We're going to inactivate the donor lymphocytes because we don't want to see the donor's immune response. We want to see the immune response, potential immune response from a recipient when we expose it to HLA antigens on the donor cells. And the HLA antigens are on donor lymphocytes. So, recipient lymphocytes and donor lymphocytes incubated together. The donor lymphocytes will not react. They're inactivated. If the recipient lymphocyte recognizes something foreign, that is, it's not HLA compatible, it's going to start to react and it's going to mount an immune response against the foreignness of the donor cells. And what happens is those lymphocytes start proliferating. They split and they separate. They split, they develop into, they split and develop into daughter cells. So you are proliferating the lymphocytes in vitro from the recipient. Now we know we can't see this happening by itself. We can't tell the difference between a donor and a recipient lymphocyte visually if we were looking and watching this. So what do? We can use a radioactive label called thymidine, and it's going to label the DNA only of cells that are proliferating. So if there was no response from the recipient lymphocyte, it didn't spot anything foreign, it didn't spot anything that it needed to react to, and it didn't proliferate, there would be no tagging by radioactive because it's only in active DNA of actively proliferating cells. However, if the recipient lymphocyte came in contact with a donor lymphocyte that had different HLA antigens on it, had different antigenicity, and it saw it and said, this is foreign. What am I going to do? I'm going to proliferate and I'm going to try and fight this battle. Those cells, while they're in proliferation, we can tag them with that radioactive dye or radioactive tag. And we will then check and see we can measure the signal of radioactivity after a period of time. So if there's a high degree of radioactivity, it means there was a lot of proliferation of lymphocytes. And remember I said at the beginning, we inactivated the donor lymphocytes. So if there was proliferation, the only cells that could be proliferating are the recipient. And what does that tell us? that tells us that if we transplant that donor cells into that recipient, there's going to be an immune response that comes with it. The immune response reacted to the lymphocytes, it's going to react to the donor cells as well. Has the same HLA, has different HLA antigens. They're two different. That'll be a non-suitable donor. So that mixed lymphocyte reaction test is really a relatively simple test to determine if a donor is likely to stimulate a graft reaction. We're going to see there's going to be different ways for us to do this, but I think it's historically it's a good understanding of what it is, and I think visually it gives us a good picture of why we do it. So that's direct recognition, direct allorecognition. Cytotoxic T cells from the recipient bind to donor cells and cause a reaction, a cytotoxic reaction. That's direct. Indirect, on the other hand, yes, we're getting to the second pathway, or the indirect pathway. This one operates via T helper cells and the recognition of peptides that were processed and presented by MHC molecules on on APCs, on antigen presenting cells of self. So the recipient antigen presenting cell does what? It uptakes a foreign antigen, processes it, and presents it to T helper cells. And if there is, so if it's a foreign cell, it presents it to T helper cells. Those naive T cells do what? They will differentiate into other cells. It may cause a cell-mediated response with cytokine release, may call for phagocytes to come and clean up the mess. It may call for natural killer cells. It may also induce B cell proliferation and production of antibody. So it could, this process, this indirect allorecognition, can mediate either an antibody production or a cell-mediated type of response. So those are the two processes that come, but the indirect is really going to be from T helper cells, not cytotoxic, and it's going to be some kind of secondary response, like I said, of cytokine production and chemokines and things like that. So this picture, again, should look really quite familiar to you. We've seen this in a number of different processes, and I think that they should look familiar and easy to follow along with at this point. So this is our direct allorecognition process, and this is our indirect allorecognition process. So what do we see? In direct, we see the cytotoxic T cells from the host, the recipient, has a T cell receptor that recognizes an antigen on the graft cell. They bind. Remember, this is a nice specific antigen and T cell receptor, and the host cytotoxic T cell, the CD8 positive T cell, releases perforins and granzymes, resulting in death of the graft cell. Very straightforward. The indirect allorecognition is one where we have the antigen presenting cell uptakes a foreign antigen, brings it into the cell. It's processed. This antigen came from grafted tissue, right? So grafted tissue is brought in. It gets processed by antigen presenting cell. The MHC does what? Brings that little teeny peptide up here. The peptide binds into the T cell receptor, and the host CD4 positive T helper cell has a number of reactions. They can release cytokines. They can end up with differentiation of cells, resulting in cell-mediated cytotoxicity or antibody-mediated reactions. So we can end up with humoral or adaptive immune response now. Cell mediated or humoral response. So looks familiar to you? I think it should be. Indirect, we see normally in chronic rejection. It's a little bit slower because, and why, how do we know that it's going to be a chronic rejection? What kinds of things are we doing here? These are not this kind of immediate reaction. This is really a longer term effect. So it's those chronic, slower rejections that we expect to see. Some of the mechanisms of direct versus indirect, as I said, the two systems, direct versus indirect, we have the difference between direct cytotoxicity of direct allorecognition, and then this kind of delayed hypersensitivity of cell-mediated or antibody-mediated. So direct versus indirect, indirect. And that's really the differences are just the mechanisms they get to there. But in the end, in both cases, you end up with damage to the grafted tissue and toxic damage that will result in rejection of the tissue. So, we don't look at rejection in terms of just direct and indirect response. We look at it in terms of kind of how fast it happens. And we saw this in hypersensitivity reactions, for example. So, the speed by which graft rejection takes place is also going to be used in our assessment of the graft rejection. So there are three major types of graft rejection in terms of timing and their mechanisms of their effector mechanisms. The first is a hyperacute response. This occurs minutes to hours after the transplant. This would be one of the ones like we saw with the ABO incompatible antibody, the preformed antibody. And in fact, hyperacute reactions do require do require preformed antibody be present. How do we get preformed antibodies that may affect the graft rejection process or the graft process? Preformed antibodies could be antibodies to ABO, could be either an incompatible blood group or something even from a prior transfusion. You haven't done blood banking yet, but there's not just ABO antigens on red blood cells. You can have, I don't want to call them minor antigens, but they're not ABO blood group. They're other blood groups. Kel, JK, Kid, Duffy. There's a slew of them with different variants within them. And you can, if I, for example, don't have a Kid antigen and I get transfused with blood cells that do have a Kid antigen on it, I'm going to develop antibody. And nothing like a good memory-filled immune system so that the next time I expose that antigen, perhaps on a tissue transplant, I have antibody that's preformed and can respond very, very quickly. It could be something though like an ABO antigen or antibody where I have anti-B antigen, anti-B antibody because I'm a type A person. So you can have preformed antibodies to ABO antigens. You can have preformed antibodies maybe from a prior transplant. And other things we saw a couple of weeks ago, how you can have preformed antibodies if you were exposed as a mother to a fetal antigen from paternal origin, and how you can, if you're exposed to a fetal antigen, you can develop an antibody to it. All of those are preformed antibodies that can, in fact, cause graft rejection. How does this work? It's a good old antigen-antibody reaction. We're going to activate complement when they bind. Also going to activate some clotting factor cascades, and thrombi form in the tissues, and ultimately result in necrosis of the donor tissue. So it's not a really common reaction anymore because transplants are really carefully paired up for ABO compatibility, and they're screened constantly for preformed HLA antibodies. So it's not as common as it used to be, but it does still happen. And again, it occurs minutes to hours, after the transplant. There's a version of this called, kind of a subversion of this called an accelerated reaction. In this case, there may be instances where there's still preformed antibody, but it's a very low level, and it's an accelerated response can occur a couple of days after the transplant, but ultimately leads to mechanisms the same as the hyperacute. It's just going to take a few days for the immune system to kick in to the point that it will cause damage to the donor tissue. The next major type of graft rejection are the acute graft rejections. These take days to months after the transplant to kick in. This is a cell-mediated response. This is one in which, kind of as expected, cytokine production responds to CD8 positive T cells, and it will cause a delayed hypersensitivity style response. And this is again in response to the identification of foreign antigens from the donor cells. So in addition to, we just saw this. I did it down. Sorry. We just saw this. This reaction. We can have a cell-mediated, or we can have antibody-mediated reactions in an acute phase graft reaction. Some immunosuppressive drugs can actually improve this outcome to some extent, but we'll see in a few minutes that immunosuppressive drugs are not without consequence. And then chronic graft rejections recur a year or more after the transplant. It too is a delayed type hypersensitivity reaction. It takes a while for you to eventually develop it, but an alloantibody is eventually developed against the donor cells. It ends up having damage to the vasculature, arteriosclerosis, some scarring, ends up narrowing the lumen, the central area of the vasculature where the blood flows, and rejections follow. We can also see chronic graft rejections when you have some particular immunosuppressive drug therapies that can actually cause a damaging response where the tissue ends up dying as well. So those are the three types, almost four, three and a half types of graft rejection responses that the host can elicit against donor cells. There is one more though, and we've talked about this really briefly today so far, and this is the graft versus host disease. This kind of turns things around a little. In graft versus host disease, there are going to be lymphocytes that are embedded within the tissue, the donor tissue that gets grafted into the host. What happens is when they leave the tissue, they notice all this foreign antigenicity in the recipient, and they mount an immune response against the recipient's own antigens, their own histocompatibility antigens, and it causes an immune response that that is against the recipient. So it comes from donor cells that sort of hitched right in with the tissue and caused an immune response against the host. We see this kind of disease, this graft versus host disease, in patients, especially ones with hematopoietic stem cell transplants, and to a lesser degree, some lung and liver transplants we can see this in. So why does this happen? We'll take a look at stem cell transplants particularly. In stem cell transplants, the recipient, their bone marrow gets wiped out by chemotherapeutic agents, and they get an infusion of donor peripheral cells that contain stem cells. But the infusion of donor stem cells doesn't just contain stem cells, but also has some, super nice, friendly, immunocompetent lymphocytes there. And a few of those mature immunocompetent lymphocytes are very good actually for the stem cell transplant. It can help the transplant to engraft into the bone marrow and other sites of extramedullary hematopoiesis. And it can also do something called, it can mediate something called the graft versus leukemia effect, or the GVL. And what that is it's essentially those healthy T cells are going to recognize malignant cells that may still exist in the host or the recipient and cause an immune response against them. So it's a nice, healthy reaction to have. That's the good news. The bad news is, these donor T cells are going to recognize the recipient cells as foreign and can release massive amounts of cytokines, cause inflammation, destruction of tissue, including things like the skin and the GI tract. It can have a really overarchingly bad response for the recipient, and we call that graft versus host disease because it's the graft responding against the host. Sort of Davy versus Goliath. In this case, it's graft versus the host. This tends to occur, 100 days or more after the transplant. It doesn't happen overnight. Again, the incidence and severity of this graft versus host disease is based on how mismatched is that HLA antigen that we were doing. We're going to see it's really important. Those, they're so antigenically diverse. You have to find a tight match, or else you end up with these reactions of the immune system. We can treat it with immunosuppressive therapies, and we can do some removal of T cells. As I said, though, T cells are really helpful in engraftment and that graft versus leukemia response. So we don't necessarily want to just be, why don't we just remove the T cells anyway? Because there's a reason. They're actually helpful in the process of the hematopoietic stem cell transplant. So question 2, in graft versus host disease, which is a true statement? Immunosuppressive therapies inhibit antibody-antigen binding. Well, no, that's not what we said. Donor cells are transfused with tissue and mount an immune response in the recipient. I think that's so. And graft versus host is an acute reaction. Well, no, we said it took 100 days, so that certainly couldn't be. So it's B. Donor lymphocytes are transfused with tissue and mount an immune response in the recipient, against the recipient, right? So, we've talked about a couple of times today how important it is that most of the time we need some sort of drug therapy to suppress those anti-graft responses of the host or the recipient. The drugs also increase, has some negative side effects, can increase the recipient's susceptibility to infection, malignancies, can have other side effects. We've already talked about some of them. So they can cause some damage to the tissue and cause a graft type reaction. So we, a chronic graft reaction rejection. So, but we do need it, and so we try and find the best one and the best fit for what we're doing. Let's take a look at a couple of them really briefly. We'll describe them and then move on. So corticosteroids are anti-inflammatory agents. They block the secretion of cytokines and other mediators of inflammation. They block the adhesion of molecules and make it so that, in general, the immune system is not functioning at its best. Long-term use, though, can result in some complications like hypertension or diabetes mellitus. So the next class of immunosuppressive agents are antimetabolites. Those can interfere with lymphocyte maturation and division. The purpose of that is so that if we're killing we're killing off the proliferating lymphocytes that may respond to HLA antigens, there's a new class of drugs called mycophenylates that are the most effective antimetabolites at this point. So antimetabolites just are designed to interfere with the maturation and division of lymphocytes. Calcineurin inhibitors like cyclosporin impair the cytokine synthesis, like some of the interleukins, interleukin 2, 3, and 4, and it blocks the growth and differentiation of T cells. So very similar to calcineurins, but in this case, instead of interfering with maturation and division, they are they're blocking growth and differentiation of T cells. So it's a slightly different function, but it's really inactivating the action of T cells. Let's see, monoclonal antibodies. I think this is we're familiar with this monoclonal antibodies at this point and their process, but in general, monoclonal antibodies interfere with either T or B cell function by binding to cell surface molecules on lymphocytes. Some of the ones we see are things like anti-CD25 is a monoclonal that's in use. Some of the issues that come up from this, though, are, we talked about how monoclonal antibodies were derived originally from mice, and so when you are using monoclonal antibodies, you can develop anti-mouse antibodies that can bind off to those monoclonal antibodies, the reagent antibodies, or the immunosuppressive antibodies, and make them ineffective. So it's not a perfect world. Polyclonal are animal-derived antibodies. They started using these years ago. They would inject the antigen into an animal, a mouse or something, and harvest their serum and take the antibodies from that and put it into the human. But in this case, so polyclonal antibodies are animal-derived antibodies, and an example is something like thioglobulin, which has a action as an anti-thymocyte, resulting in a depletion of thymocytes and ultimately lymphocytes. So we're reducing the population of lymphocytes in the recipient. Problem with this, we saw a couple of weeks ago the development of serum sickness when you're using animal-derived antibodies. So again, not necessarily where we want to head, but you got to do what you got to do, I guess. So as you can imagine, HLA matching of transplant candidates, so we're avoiding these problems. The best match between HLA, between the candidate and the donor, is really the most important thing we can do to reduce the chances of graft rejection. The typing, HLA typing, involves identification of those HLA antigens or the genes in the transplant candidate or the donor or both, for both solid organ and hematopoietic stem cell transplants. So we're going to want to see what the antigenicity is between donor and recipient. That's our ultimate goal here. We're going to do a couple of different tests to look for histocompatibility testing. So typing, we're going to do some phenotypic typing or genotypic typing. And the test we're going to use for phenotyping is called complement-dependent cytotoxicity testing, or CDC testing, and molecular testing for genotypic screens. We also can do, when we're doing HLA typing, we don't have to just look at the antigenicity. We can look at the antibodies, and we can screen for those. We can identify antibodies that the recipient or the donor may have, and we can cross-match them. And we saw just a moment ago when we were talking about graft versus host how important it is for us to see if the donor has antibodies as well that may interfere with this. So it's not just antibodies of the recipient, but we also want to look at the recipient as well. So let's look at the phenotyping process. So we're going to do phenotyping processing for HLA antigens in a candidate and a donor. And we're going to look at their specific HLA phenotypes of the groups of HLA A, B, and C. We're going to use a test called complement-dependent cytotoxicity, and we're going to look at that in just a second. We have an illustration on the next slide. But in this moment, let me just tell you that this test gets graded on a scale of 0 to 8. 0 means the test is unreadable. The cells are not valuable. And then we're going to grade it based on how many cells that we're looking at are dead. I'm going to tell you how we identify them as dead or not. But a negative test is going to show that very few of the cells that we are testing are dead. A positive test, that is in identifying the phenotyping of the antigens on this cell, is going to be a lot of cell death. Let's look at why this is the case. And then obviously there's gradations everywhere in between negative, doubtful negative, which is a 2, a weak positive is a 4, a positive is a 6, and a strong positive is an 8. So let's look at how we get to that point. So this is, again, an illustration from your book. And this test is done in a microtiter plate. So each one, this is one test. This is a series of pictures for one test. This would represent one well in the microtiter plate. We don't do things one at a time in the lab. This process, so as I said, it uses separate microtiter plates are used for a candidate, transplant candidate, and all of the potential donors. Maybe it's a family of donors or something, but it's all of the potential donors. So every person that gets tested here gets their own microtiter plate. No mixing and matching. Each well of the plate is going to contain antibodies in it, and this is going to be reagent antibodies. And this antibody is going to be specific to one of the HLA antigens in question. So remember, we were going to look primarily at A, B, and C, right? We're going to look at we're going to determine the phenotype of A, B, and C, and we're going to compare them. So we have the specific phenotypic information in a series of antibodies that gets added to the test. So those are reagent antibodies. The cells are either donor or recipient cells. Remember, we're going to set up a microtiter plate of donor cells. We're going to set up a microtiter plate of recipient cells. And what we're looking for is the cells, do they have, the antigen on the cell that we're looking for. So the patient, whatever the patient is, whether it's the donor or the recipient, the patient cells are incubated with the reagent antisera for all different class 1 and class 2 HLA antibodies. So we're going to have multiple antibodies we're looking for. And we're going to see if we get an antigen-antibody binding. We know if in a certain test well, we know what the reagent antibody is. If we end up getting a reaction there, then we know that the patient has the corresponding antigen to the reagent antibody, right? It's like all of our tests that are antigen antibody based. Nothing new here. What gets a little funky is, so we have let's say this is a donor, let's pretend this is a donor test. We have donor cells that have antigens, HLA antigens on it. Not all of them do. This one, if this one has antigens on it, this cell will represent what happens if there's no antigens on it. So if there's the antigen that matches this reagent antibody, they bind. The antibody binds to the antigen. And so when two antibodies bind close together, if we add complement in, so this is a reagent complement, and this complement is actually harvested from donations, and we harvest complement and use that. So we have complement here. What happens? So when you have an antibody binding, and you have two of them nearby that complement can span, they have to be next to each other. Complement binds to this, and what happens to this cell? The MAC attack happens, and this cell is going to what? It's going to lyse. It's going to die. So would we see this reaction by itself? No. What happens? So we know we don't see sensitization, which is an antibody binding to an antigen, and we don't necessarily see this by itself. What do we do then? So we're going to wash this again. So we add antibody, let it incubate, we wash it, add complement, let it incubate, wash it. How are we going to detect if this reaction took place or if no reaction took place? We're going to add in a dye into the test system. And the dye is going to be what's called a supervital dye or a supervital stain. And that's a stain that only gets taken up by live cells. If the cells are dead, they don't stain. So cells that have been lysed by complement will not stain. Cells that are not lysed will not stain. So we're going to use a couple of different kinds of stain depending on the test kit. One could be something like eosin, or one could be trypan blue. In this case, this appears that they use trypan blue here. So we're going to go with that. So we get through this whole thing. This is dead. It didn't lyse. No stain was taken up in this cell. This should have been a little bluer or something to demonstrate that I took up the blue stain, but they didn't do that. So I can only work with what I got. So this, remember we looked at the scoring? A 0 is 0 to 10% of the cells were dead, and it's negative. So if we added trypan blue to this, and all the cells took up the stain, meant none of these were dead. So this is 0% cells were dead. So this test is negative. This patient's cells did not have the antigen that matched this antibody. So if we were looking for a specific phenotype of an antigen, of an HLA antigen, we know that this antigen this cell did not have it because this reaction never took place. This reaction never took place where it killed the cell. None of the cells were dead. No complement. That meant there was no antigen-antibody reaction. Does that make sense? I hope so. In the next test well that we're looking at, roughly half, I think they could have done a little bit better with their numerical counting because they really kind of maybe shouldn't have made this a 4, but they did. In this case, it's actually technically just over 51%. It should have been a 6, but 6 out of 13 of the cells stained blue. That meant 7 did not. 7 out of 13 is just over half. But let's pretend like they didn't do that to us. And 50% of the cells were dead. So it's a weak positive. It's a weakly positive reaction. There's some cells there that have this antigen on it, about half. Some cells do not. These cells have the antigen. They didn't take up the stain. These 6 cells did not have the antigen, and so they didn't react. This third one, none of these cells took up the stain, so they're 100% dead. If there's 100% cell death, anywhere between 81 and 100% cell death, it's a strong positive. This tells me that every single one of the cells that we tested from this patient had the antigen in question on this cell surface that we had in mind. We're going to do that, and we're going to compare. So we're going to do this to the donor cells, to the recipient cells, and we're going to do it to the donor cells. And we're going to get the best match where multiple donors are going to be tested so that they all have the same antigenicity. So that when we put the recipient transplant into the donor is not going to have antibodies to it because they have the same antigen. Does that make sense? It's really important that we get there. So if you don't get this, ask me about this. Shoot me an email or a text or an online post, and we'll discuss it. But this is the basis of HLA typing that makes us know that this is going to be a good match or a bad match. We want to make sure that we only find patient and donors that have the best, the best match of HLA antigens on their genetic material. So you'll do this with a bunch of different antibodies, and you're going to find the ones that have the best match on all the microtiter plates. And again, you're going to be doing one recipient and many donors here. So we'll look from there. So, complement testing, there is a place for it, but now, that did phenotyping. Now we can use PCR and look at, really we can amplify the variants, the alleles of the donor and the recipient, and analyze the DNA in great detail. We can determine the specific allele groups of the donor and the recipient, and for a match. So we have a really good opportunity for identification. The picture on the right just tells us the nomenclature numbering of this. And each one of those when we're doing the typing, we'll have the gene locus. The locus is on HLA A. The allele group is the O3 group. The allele number is O1. The synonymous variation, non-coding variation, the variation in expression, all go into this large genotyping number. And we're going to see this used in just a few minutes as well. PCR clearly has advantages over CDC. Obviously, we say it has a higher resolution of genotyping because, honestly, it gives us better information compared to what we need. So we'll be able to do, genetic identification rather than a slightly more simplified version that we saw with the CDC testing. One of the things, we don't require viable cells. The cells can be dead. So that's good news. We don't have to worry about that because we're really just looking at the DNA. And as I said, we're not using human donors like we did in the CDC testing. So chemical synthesis means we're getting some consistency on our reagents. And obviously it's a faster test. It's not as prone to, operator error, all those things. A couple of different molecular methods we're doing. PCR SSP, which is sequence-specific polymerase chain reaction. That's the typical PCR that we would expect, and we'll see that in just a second. The next is PCR SSOP, which is PCR with sequence-specific oligonucleotide probe hybridization, and we'll see that in a second. And then SBT, which is a sequence-based typing. The only other thing that I guess I want to say about this is this M right here of the variation expression is where we see that this allele, this allele does not have an expression at that point. It doesn't have a variation in expression. So if we were looking to see if there were variants in HLA antigen, it would be classified, and when we put the nomenclature out there, that N at the end means that there's no variant in there on the cell surface. You can have variants of the same allele, and they're going to react differently. So, question 3. True or false? In complement-dependent cytotoxicity testing, I'm missing a word. Maybe it says, I don't know. Does complement-dependent cytotoxicity testing combine reagent cells, reagent antibody, and donor complement to complete the test? Let's see. So we need either donor cells or recipient cells. So we already know that's wrong. We do have reagent antibody, and we don't get our complement from the donor. We get it also from a reagent test kit. So this answer is false. The first PCR method we're going to look at is the one that we looked at last week, the SSP, the sequence-specific PCR. This is what we typically think of when we think of PCR, where only the primer pairs that bind perfectly with the target gene will amplify in the test, and then we detect the amplification and separate it by gel electrophoresis. By knowing which primer was amplified, that's how we identify the genotype. So we know what our target was, and because we know the target, we know what the genotype of this particular HLA antigen is going to be. So, but it tests just a small piece, right? It's only going to test a little small piece. The principles of sequence-specific oligonucleotide PCR again uses a microtiter plate where DNA is amplified by PCR, but instead of amplifying one gene, we amplify all the gene variables or alleles at once. We don't have to do things sort of one at a time. We can incubate the amplified DNA then on a membrane that contains these probes. So we amplify the DNA altogether. We amplify one gene and do that. And then we're going to look for the match, not based on amplification on the match to the probe like we did here. Here we had a probe we were looking for, and we looked to see if we amplified the corresponding piece of DNA in the patient. In this case, we amplified all the DNA, and then we laid it over a membrane that had a probe here. So here's where we look for the match of the patient DNA to the probe, and it's detected with a color signal. We're going to look for a color signal in the well, and that's going to signal that the HLA alleles of the patient match the probe in question. So we know what it is, we can identify the probe, and we can identify what the patient, whether the patient had the HLA antigen. And then the sequence-based typing is one in which we amplify the gene of interest, and instead of doing just little, small pieces, we can complete the sequencing reaction using something called D-deoxynucleotides with fluorescence staining the labeled terminators. And what we do is we sequence the entire gene and we compare it to a database of HLA sequences. And it's literally a database of sequences. This detects all the nucleotides of the target rather than just those little small sequences like we see in other PCR-based tests. And so it's a very, very specific identification of the HLA antigens and any variants of them against a database. And so you do this for the patient, you do this for the donor, and you get a very good match on whether or not they have the same HLA antigens. So this is a really a test that's actually becoming put more and more into use. Question 4, which would be an appropriate choice for detecting complete nucleotides of targets? I think we just talked about this one. It was SBT, right? It was. We're coming up close to the end here. So we've talked an awful lot about testing for HLA antigen. Well, you know what? Antibodies are important too. And we know that the recipient, under certain circumstances, can develop antibodies, HLA antibodies. We talked about it earlier. They can develop antibodies after multiple blood transfusions, maybe a prior organ transplant, maybe even something like we saw last week or the week before when we talked about pregnant women developing antibodies to fetal antigens that were derived from the paternal inheritance. So what we do know is, we've figured out that, what HLA antigens people have. Let's find out if they have any antibodies too. And we're going to do a cross match to do this. It's really important we detect if there are antibodies, because donors with the HLA antigen will react with a patient with the recipient antibodies in a transplant candidate. And if you have that reaction in vitro, we certainly don't want to pop an organ into someone. We're going to see a couple of different methods that we can use for identification of antibodies: CDC, ELISA, and flow cytometry. And then we're also going to do one more thing in our final antibody screening and identification / cross-match process. Is we're going to look and see if there's any preformed pre-transplant antibodies that might cause a reaction. Patients get tested for this every month prior to receiving a transplant. And if by any chance HLA antibodies are detected, they check and see if the donor has the antigen, and if they do, that candidate is eliminated from contention for donation. So how are we going to do some testing for antibodies? We're going to do a variation on the CDC method, where we are instead, if you remember, you go back to that CDC method, in the method where we were testing for antigens. In this case, we're going to use patient serum instead of patient cells, and we're going to incubate it with reagent cells, reagent lymphocytes, 30 to 60 different kinds of lymphocytes containing known HLA antigens. So we're going to have reagent lymphocytes with antigen on them, 30 to 60 of them in the test in each and an individual test well. And we're going to add patient serum. We're going to question if there's an antibody there. We're going to let it incubate. We're going to wash. We're going to add complement. And then we're going to stain and evaluate the same way we did for cell death. We have an opportunity. We can use some anti-human antiglobulin. Did I do a picture of it here? Similar to this, but an antibody to an antibody. I didn't do an extra picture. That can increase the test sensitivity, just because it is, you have one antibody and you can be looking at it that way. That maybe is going to be very specific for what we're looking for. But instead of the way we looked at it before with cell death, we're actually going to be looking at what's called the percent PRA, which is the proportion of lymphocytes in the panel that are killed by the patient's serum. So we don't score it on that 0 to 8, but we're going to do a percentage of lymphocytes that are killed by complement. So that's the HLA antibody. Remember, in CDC testing, cell death means there was an antibody so it's not suitable for transplant. You can also, in this test method, when we're reporting HLA typing for antibodies, you can calculate, much like we do in the blood bank. You'll see we can do something called a CPRA test, which is the calculated PRA test. Instead of doing a percentage of antibody detected, we can do a calculation that will determine what percentage of the potential donor pool have the antigen for which our recipient has the antibody. That's helpful to figure out how big of a donor pool you need to look at. If you have a low incidence antigen, not very many people have it. If the patient has an antibody, you're not going to have to be scouring through high numbers of organ donors before you find someone who's compatible. If you have what's called a high incidence antigen that the patient, or the recipient, I should say, has an antibody to, so the donor has a high incident antigen and the recipient has the antibody to it, high donor recipient, what did I just say? I can't think. High incidence antigens means that a large portion of the population have that antigen. So if you have a population that most people have an antigen for which the patient has developed an antibody, it's very hard to find a donor for those people. So you would do a lot of screening to find someone that would be compatible. The next test we're going to do is ELISA testing. We talked about this 2 weeks ago, so I'm not going to spend time on it, but in this case we're going to use reagent HLA antigens bound into a microtiter plate in patient serums, and we're going to look for that enzyme-linked assay testing. And then flow cytometry is the last one. In this case, we're going to have purified HLA antigen that we adsorb onto latex beads. So we're going to force it onto latex beads. And we're going to mix it with patient serum. That alone, remember, that might be sensitization, an antibody-antigen reaction. We can't see that by itself. So we have to do something. We have to add a tag. And we add a tag in the form of an anti-human antiglobulin that is fluorescently tagged. We're going to wash it again, and we're going to look for fluorescence on a flow cytometer. So this is the latex bead with antigen in question. So in this case, we were looking for HLA-A11 antigen. So we're going to look and see, and in this case, the patient bound to it. So we know the patient has the antibody, but again, without this fluorescently labeled antibody, we wouldn't know it. This is just a sensitized cell. We need to add something that's got a signal on it. And in this case, we're adding a fluorescent signal to an anti-human antiglobulin. Binds to the Fc portion of the patient antibody, which bound to the antigen HLA-A11. So look for fluorescence. Bam, you're done. If you look in your book under this illustration in chapter 16, you'll see that there are, it was demonstrating you how there's low, mid, and high fluorescence built into this process. So you'll see they map out in different places, even though you may have multiple things with antigens that might bind to it. So you'll see that they have developed a way to determine that. And then I think finally, yes. So we talked about this. We talked about HLA antibodies. We talked about the presence of antibodies, the presence of antigen. Now we've detected that they're there. What do we do? If a patient has an antibody and the donor has been identified as a candidate, we're going to do another CDC test to confirm that the patient doesn't have the antibody to the donor. No, a donor recipient CDC test is done to confirm the absence of donor specific antibody. We want to make sure that the recipient does not have antibody to the donor antigens. If there's any screening test that indicates that the donor and the recipient have a corresponding antigen antibody, we don't do that. We eliminate that donor from the list of potential candidates. Those preformed pre-transplant antibodies are the ones that are going to cause those hyperacute graft reactions. This is why we do it. We can't have that happen and having the graft just rejected out of hand. So, and the way that we do this specific CDC test for this antibody cross-match, this is our last HLA testing. We tested for antibodies, we tested for antigens. Now we're going to test and see the patient and the recipient together and see what happens when we put them together in a CDC test. So we would have a test well on a microtiter plate where we incubate T cells from the donor, or donor T and B lymphocytes from the donor with recipient serum. And then we incubate it, we add complement, we add dye, and we look for cell death. So if we see cell death, we know that very specifically the recipient has antibodies to that donor, to their HLA antigens, and we absolutely cannot transplant that. There's no amount of immunosuppressant drugs that are going to help maintain that graft for a long period of time. So we are not going to transplant that. So that's HLA antibody cross-matching. Very similar to what we do in blood banking before we do our final test before we do a transfusion of red blood cells. We put the patient and the donor together and make sure nothing happens. No reaction. We don't want any reaction. That's it. Oh, one last question. True or false? The Mendelian pattern of HLA inheritance suggests that siblings are generally good organ donors. I think that's true. 75% have either full identity or haploidentity. So I think that's a good odd with siblings. And that's it. Thanks so much. I'll talk to you soon. Bye.